Hey everyone, welcome to Pen and Paper. Today topic is Krebs cycle, which is also known as Tricarboxylic acid cycle or Citric acid cycle. This cycle was explained by Sir Hans Adolf Kreb. It occurs in the matrix of mitochondria in eukaryotes and cytosol in case of prokaryotes. So let's discuss Krebs cycle in detail. The cycle does not start by its own. It requires a molecule which is acetyl CoA, and acetyl CoA is obtained from oxidation of pyruvic acid. Pyruvic acid, it is the end product of glycolysis. So here one glucose molecule, it break down and form two molecule of pyruvic acid. And the pathway is called glycolysis that convert the glucose into pyruvic acid or pyruvate. Now the pyruvic acid, it undergo oxidation by a complex which is called pyruvate dehydrogenase complex, and here coenzyme A enter the reaction and NAD positive convert into NADH. Also, one molecule of CO2 is released here. So, pyruvic acid, it is 3 carbon compound each, and upon oxidation it will form acetyl CoA. So, acetyl CoA, which is 2 carbon compound, it is the entered molecule of the Krebs cycle. So, it will enter the cycle, and receptor for the acetyl CoA, it is oxaloacetic acid, which is 4 carbon compound. So in the presence of water, acetyl CoA and oxaloacetic acid, it will combine and form a stable compound which is citric acid or citrate. So it is of 6 carbon compound, and here CoA is released. So the enzyme that catalyzes this reaction is called citrate synthase. And this step is known as condensation. Now the citric acid or citrate, it reorganizes and it will form cis-aconitate, which is again 6 carbon compound, and here water is released. Water is released here, and the enzyme that catalyzes this step is called aconitase. So this step is known as dehydration because water is removed here, and this step is called condensation, and this is dehydration process. Step. Next, cis-aconitate further reorganize and form isocitrate, which is 6 carbon compound. So it will form isocitrate, and it is 6 carbon compound. So here water is added. So this step is called hydration, and the enzyme that catalyzes this step is same, that is aconitase. And isocitrate further convert into alpha ketoglutarate. Which is 5 carbon compound. So there is a reduction of 1 carbon, that is from isocitrate, which is 6 carbon, to form alpha ketoglutarate, which is 5 carbon compound. And here one molecule of CO2 is released, and NAD positive convert into NADH. The enzyme that catalyzes the reaction is isocitrate dehydrogenase, and this step is known as oxidative decarboxylation, as CO2 is being released here and there is a reduction of carbon. So this step is oxidative decarboxylation. Now alpha ketoglutarate further convert into succinyl CoA. So it will further convert into succinyl CoA, which is 4 carbon compound, and again here is reduction of a carbon in the form of CO2, and NAD positive gets converted into NADH, and here CoA enzyme also enter the reaction. So this step or reaction is catalyzed by alpha ketoglutarate dehydrogenase. So succinyl CoA is formed, which is 4 carbon compound, and this step is also known as oxidative decarboxylation. Further, succinyl CoA, it will form succinate, which is 4 carbon compound, and this step is known as substrate level phosphorylation. So succinate will form here, which is 4 carbon compound, and here CoA enzyme is released and GDP gets converted into GTP. GDP gets converted into GTP. The enzyme that catalyzes the reaction is succinate thiokinase, and this step is known as substrate level phosphorylation. So enzyme is succinate thiokinase. The cycle will continue and succinate further convert into fumarate, which is 4 carbon compound fumarate, and it is 4 carbon compound. So here FAD positive gets convert into FADH2, and the enzyme that catalyzes this reaction is succinate dehydrogenase. Further, fumarate convert into malate, which is 4 carbon compound malate, which is 4 carbon compound, and here water is added. So the enzyme is fumarase that convert the fumarate into malate. So this step is known as hydration, as water is added here. Now malate will further convert into oxaloacetic acid and cycle will continue. Here NAD positive gets convert into NADH, and the enzyme that catalyzes this step is malate dehydrogenase, and this step is known as dehydrogenation. So this is all about Krebs cycle that contain 8 steps, and each of the step is catalyzed by particular enzyme. So this is step 1, step 2, which is further categorized into 2 parts, step 3, step 4, step 5, step 6, step 7, and step 8. So here energy is formed in this step, 3rd step, 4th, 5th, 6th, and 8th steps. Thanks for watching, and